Dear students, today we will discuss the formation of bilirubin. As you know that the lifespan of RBC is 120 days. As well as you also know that in different anemias there are the formation of abnormal RBCs. So these abnormal RBCs are broken down. Now the main area where the destruction of RBCs occur is the spleen. In the spleen, macrophages are the main cells in which the breakdown of these RBC occur. Other than spleen, the destruction of RBCs also occur in the Kupffer cells of the liver and the macrophages of the bone marrow. Now consider this is a macrophage. As a macrophage engulfs in RBC, it is broken down into hemoglobin. The hemoglobin release here is converted into two parts. One is the globin and the other is heme part. Now the globin, as you know that it is a protein portion, is further converted into amino acid and it forms the pool of amino acid in the plasma. It goes out to the plasma. Now the heme portion consists of further two parts, the porphyrin and the iron portion. Now this is broken down into the iron and the porphyrin. The iron goes to the plasma and form the pool of iron in the plasma, while the porphyrin is converted with the help of heme oxygenase into biliverdin. The biliverdin, in the presence of biliverdin reductase enzyme, is reduced into the bilirubin. This form of bilirubin which is produced is called unconjugated bilirubin. It is also called indirect bilirubin, or it is also called water insoluble, as it is not soluble in water. Now this bilirubin comes out from the macrophages into the plasma and is temporarily conjugated with, or we can say bound with, the serum protein called albumin. Now due to this albumin, they become water soluble and are transported to the hepatocytes. Moreover, the function of the binding to albumin is that it prevents it from entering to the tissue. Now as these bilirubin are transported to the hepatocytes, in the hepatocytes the bilirubin is combined with two molecules of glucuronic acid with the presence of enzyme called glucuronyltransferase. The end product form is bilirubin diglucuronide. Now it is water soluble. It is called the direct bilirubin. It is called the conjugated bilirubin. Now it is excreted through bile into the intestine. In the intestine, bacterial destruction causes this bilirubin to be converted into urobilinogen. The urobilinogen, small portion of urobilinogen is reabsorbed from the intestine and then moves to the kidney. In the kidney, it is converted into urobilin and is excreted in the urine, which gives a characteristic yellow coloration, light pale yellow coloration to the urine. The remaining portion, which is the major portion of the urobilinogen, is converted into stercobilin. Stercobilin is brown in color, which gives a brown coloration to the feces and is excreted in the feces.